So this is the second installment of the biochemistry of the coronaviruses. And today I want to talk a little bit about the coronavirus life cycle, and specifically I'll call it the biochemical life cycle. I'm not a virologist, so I'm not an expert at understanding how viruses exist outside the cell. But I do understand a little bit of the biochemistry of how viruses use our cell resources to make more of themselves. As I mentioned before, viruses are not living, and so they cannot copy themselves or make more of themselves outside of our cells. They need resources inside our cells to make more and infect more cells. And so I like this figure. As I mentioned before, coronaviruses are not a new thing. And so this is from a publication that was from 2006, but it's still very much accurate. But it may look complicated, but it's actually one of the less complicated versions of this type of figure. And I wanted to walk through some of this information here. So the first thing that happens is the virus finds cells that have specific receptors on the outside of them. And so the halo or the crown that's around the virus, those are proteins, and those proteins match up with the receptors. And so let's look a little bit at the structure of those proteins and receptors, because this was a huge biochemical feat to get the structures of these published so quickly. So here is a picture of the spike protein. This is the protein that's on the outside of the virus that causes that crown. And so there were actually two different publications. One was published in the journal Science and the other in the journal Cell. These are both huge journals in basic chemistry, biochemistry, scientific research. And so I also wanted to pull up the structure in 3 dimensions. So these are figures from the paper that describes these, but I wanted to show you what these look like kind of moving around in 3 dimensions. So this is a viewer. It's from the protein database. People who solve crystal structures of proteins publish their structures here. And so here you can see the 3-dimensional view of the protein. I'm moving it around with my cursor so you can see that it has a 3-dimensional shape. We can move it up and down. This little orange piece on the top is the piece that fits into the receptors on our cells. And so the bottom of this would be in the viral membrane, and then this orange piece is going to attach to our cells. And so that's the spike protein. I should also mention that on this figure you see these little dark blue blobs. These are different sugar molecules that are attached to the spike protein. So people are looking into seeing how sugar affects the binding of the spike protein to our receptors. So then let's look briefly at the receptors. And so this is the specific receptor on some of our cells. Again, these are recently published. I'll pull up the 3-dimensional view. So here's the 3-dimensional view. You can see that it you can't see that right now. You can see that it kind of has a mirror image. It's a dimer. And here are the screen and yellow is the part on the spike protein that's binding this receptor. So here I'm moving this around in 3 dimensions. Again, I'm not sure if it's showing up. There. It's kind of moving now. So there you can see it in 3 dimensions. I also wanted to point out that this receptor has a normal role So again, the spike protein's binding this receptor. The receptor's there because we need it for other things. And so this is from the paper that published the structure of the receptor, and it talks about kind of the importance of this receptor in normal roles. So physiological role just means a normal role. What I find interesting is what happens once the virus attaches and gets into the cell. So this is the rest of the life cycle that I find interesting, particularly because it deals with RNA. So if you remember the central dogma molecular biology, I believe I talked about it in the first installment, DNA gets made into RNA, and this process is called transcription. However, the coronavirus does not have any DNA, nor does it ever make DNA, and so this process is not important to the coronavirus life cycle. But RNA gets made into proteins, and this process is called translation. And so this process in our bodies takes up a significant amount of the energy of our cells. I've read anywhere between 70 to 90% of our cells' energy goes into this translation process. The main cellular machinery that does this translation is called the ribosome, and it's shown in this figure as these two blobs. Ribosomes are often shown like this. It's somewhat similar to their structure, but these ribosomes are the main machinery that makes the proteins. And so coronaviruses do not have their own ribosomes or any of the resources needed to perform translation. Instead, once they enter our cells, they have disguised their RNA. It makes it sound like they're alive, but they're not. The RNA looks like RNA that comes from our cells. And so once the ribosomes inside our cells see this RNA, they start translating it because they are programmed to translate any RNA that looks like the right RNA. And this coronavirus RNA looks like it is the correct RNA. The first thing that it translates is this protein called replicase. Anytime you see something in an ACE, it's usually a protein, and specifically a protein that does catalytic function. So it does important functions in the cell. The function of this replicase, so that's the first protein that's made, is to make more RNA. And so normally in our cells, we do not have anything that does this replicase function. We don't have a way to copy RNA to make more RNA. So this is unique to the coronavirus. This replicase takes RNA and just copies it. And so we get a lot of different RNA pieces. Some of them are the full-length RNA, some are smaller pieces of RNA, and I actually find this process extremely intriguing. There are certain things that are encoded within this just single piece of RNA that makes all of these different shortened pieces of RNA. Again, I find it super fascinating, but it's way complicated. If you're interested in it, I would gladly talk about it because I think it's really cool. But then all of these RNA pieces also get translated to make proteins. It makes the S protein, the E, M, N proteins. The S protein is that spike protein. And so because all of this translation is occurring to make proteins that are not our own proteins, it is depleting our cells of the resources they need to make their own proteins. And instead, making enough proteins for the virus to make more of itself. So once there's a buildup of these proteins, new viral particles can be assembled and released and then infect other cells. And so this is the kind of overview of the life cycle of the coronavirus. I wanted to go over this because in future installments of the biochemistry of the coronavirus, I would like to talk about how some of the testing is performed and different treatments that are being that are running in clinical trials right now, and it helps to kind of have a basic overview of what's happening inside our cells and how the coronavirus infects and hijacks our cells, and the unique parts of the coronavirus in order to understand how some of that works. So I hope this was informative, and I hope to post another one soon.